Assalamualaikum. Fatin.
Waalaikumsalam.
Fatin, you have an unmute nya.
Waalaikumsalam, Doktor.
Okay. So this one Fatin first kan? Or
No.
other?
Other
Oh
people.
bukan. Oh
Bukan,
other
bukan.
people.
Yes. Other people.
Okay. So yang present kena ini lah, kena open video and unmute sekali. Silahkan satu yang kitab Al-Fatihah. Bismillahirrahmanirrahim. Alhamdulillah. Iya kan Okay, who is the first presenter? Aisyah. Aisyah belum unmute nya. Unmute dulu. I cannot hear you, Aisyah. Ada lagi suara.
Assalamualaikum. Can you hear me?
Slow, not clear.
Ada suara tak? I don't hear anything.
Cannot hear. Macam jauh.
Sekarang dengar tak?
Kak Malin tak dengar.
Kenapa tak dengar?
Cuba cakap kuat sikit.
Okay. Hello. Hello.
Dengar.
Dengar.
Tapi tak kuat.
Dengar. Boleh ke kuat lagi? Ke tak boleh dah?
Okay. Sekarang kuat tak?
Oh, okey lah tapi jauh-jauh. Dia macam jauh-jauh.
Jauh.
Okay, I think we can start lah. Coba try Aisyah. Amalin ya. Hilang lagi suara tu.
Tak dengar langsung.
Kalau sekarang better tak?
Ya, okey.
No need microphone lah. Just direct to your laptop ya.
Bismillahirrahmanirrahim. Assalamualaikum and a very good morning. I bid to Dr. Taufiq and fellow friends. My name is Nia Shamari, and inshallah today we will be presenting seminar 11 which on the topic of genetics. So in this topic of genetics, we will discuss on first molecular basis of genetics, second autosomal disorder, third sex-linked disorder, and fourth chromosomal disorder. Okay, next. Okay, so first and foremost, before we go to the disease of genetics, we will discuss on the molecular basis of genetics. Okay, next. For this basis molecular basis of genetic, we will cover about the introduction, chromosome, genes and allele, DNA, mutation, and also importance of molecular genetics. Okay, next. Okay, for introduction, we will look into the definition of several terms in genetic. The first one is genetics. Genetics is the study of heredity and of the mechanisms by which genetic factors are transmitted from one generation to the next. Any dysfunctional gene or gene mutation can cause illness and can be passed from parents to children. Okay. The second one is genomic. Genomic is the study of the structure and action of the genome, which is the sum total of genetic material present in an organism. Okay, the main difference between genomics and genetics is that genetic focus on the functioning and composition of the single gene, whereas genomics address all genes and their interrelationship in order to identify their combined influence on the growth and development of the organism. Okay, the third one is molecular biology. Molecular biology is the field of biology that studies the composition, structure, and interactions of cellular molecules such as nucleic acids and proteins that carry out the biological processes essential for the cell's function and maintenance. Okay, and the last one is molecular genetics. Molecular genetics is the field of biology that studies the structure and function of genes at a molecular level. So in this topic, we will focus more on molecular genetics. Okay, next. Okay, as we all know that our body is made up of trillions of cells. They provide structure for the body, take in nutrients from food, convert those nutrients into energy, and carry out specialized functions. So in the cells, it contains nucleus. In nucleus, we have chromosome. And in the chromosome, it contains DNA. And lastly, a segment of DNA, it gives a structure called genes, which contain hereditary materials. Okay, next. So first of all, we need to know about the chromosome. What is chromosome? In the nucleus of each cell, the DNA molecule is packaged into thread-like structure called as chromosome. So each chromosome is made up of DNA, which is tightly coiled many times around the proteins we call as histones that support its structure. This DNA carries genetic information in the form of gene. So each chromosome consists of two identical chromatids and a centromere. We can look at the figure of chromosome here. The constriction point is called centromere, which divide the chromosome into two sections or arms. The short arm of the chromosome is labeled as P arm, and the long arm of the chromosome is labeled as Q arm. The location of the centromere on each chromosome will give the chromosome its characteristic shape and can be used to help describe the location of specific gene. Okay, next. In humans, each cell normally contains 23 pairs of chromosomes for a total of 46. 22 of these pairs called as autosome, which look the same in both males and females. The 23rd pair is the sex chromosome. It is different between males and females. Females have 2 copies of X chromosome, while males have 1 X and 1 Y chromosome. If you look at the right side, this is the karyotype for human chromosome. The chromosomes are lined up in pairs. The 22 autosomes are arranged and numbered by size. The other 2 chromosomes, X and Y, are the sex chromosomes. Okay, next. Okay, next we will look at the genes and allele. Okay, next. Okay, a gene is the basic physical and functional unit of heredity. Genes is a segment of DNA molecule that codes for a particular protein or particularly known as trait. For example, we have specific genes for eye color. We have HER2, HERC2, and OCA2. And we have MC1R gene, which is specific for hair color. Okay, next is allele. Allele are forms of the same gene found on the same locus on homologous chromosome, which control the same characteristics. It has small differences in their sequence of DNA base, in their sequence of DNA bases, and these small differences will contribute to each person's unique physical features. Okay, for example, if we look at the diagram, for example, genes for eye color, there is allele for blue eyes, which is recessive allele, and allele for brown eyes, which is dominant. In individual A, he has heterozygous allele, blue and brown eyes. So individual A will have brown eyes since brown eyes allele is the dominant one. In individual B, he has homozygous allele for brown eyes. So individual B will definitely have brown eyes. In individual C, he has homozygous allele for blue eyes. So even it is recessive allele because since it has both allele for blue eyes, thus he will have blue eyes. Okay, next, we go to DNA. Okay, DNA, or known as deoxyribonucleic acid, is the hereditary material in humans and almost all other organisms. DNA is a polymer of nucleotides covalently linked by 3' to 5' phosphodiester bonds. Most of DNA is related in the cell nucleus, where it is called as nuclear DNA. But a small amount of DNA can also be found in mitochondria, where it is called as mitochondrial DNA or mtDNA. Okay, DNA consists of 3 basic components, which are deoxyribose sugar, phosphate group, and nitrogenous base, which are adenine, guanine, cytosine, and thymine. The information in DNA is stored as a code made in these 4 nitrogenous bases. Human DNA consists of about 3 billion bases, and more than 99% of those bases are the same in all people. Okay, the order or sequence of these bases will determine the information available for building and maintaining an organism. Okay, these DNA bases will pair up with each other, which is A with T and C with G, to form units called base pairs. Each base is also attached to a sugar molecule and a phosphate molecule. Together, a base, sugar, and phosphate are called as nucleotide. Okay, these nucleotides are arranged in 2 long strands that form a spiral called as double helix. Okay, next is the function of DNA. Okay, DNA is responsible in long-term storing and retrieving of information. DNA also important in performing 2 main vital processes, which are replication and transcription. Okay, for replication, it is when the genetic information processed by the parents are transferred to the progeny, whether daughter cells or offspring. An important property of DNA is that it can replicate or make copies of itself. Each strand of DNA in the double helix can serve as a pattern for duplicating the sequence of bases. This is critical when cells divide because each new cell needs to have an exact copy of the DNA present in the old cell. Okay, the second one is transcription. The process by which the information in a strand of DNA is copied into a new molecule of messenger RNA, which is mRNA. This is the source of information used for the synthesis of all protein molecules of the cell and organism. Okay, next is the process of, we look at this diagram. This is the process of DNA and RNA and protein. DNA will undergo replication in order to make copies of itself. Then DNA also will undergo transcription to produce RNA. Then RNA will undergo translation for the synthesis of proteins. Okay, proteins are the executors of all actions and functions of life, So if there is any change in DNA, it will affect the protein. Okay, next. Okay, then we look at the RNA transcription. Okay, in order to change from DNA to RNA, DNA will undergo transcription. Okay, the synthesis of RNA molecules using DNA strands as the template, then the genetic information can be transferred from DNA to RNA. We take a look at the diagram. The first one, RNA polymerase will bind to promoter at the template strand of DNA to unwind the DNA. Second, there will be initiation of RNA synthesis. Third, there will be elongation of RNA, which is RNA polymerase will move along the template strand and synthesize the mRNA molecule. Okay, the fourth one, this will be proceeded until they reach termination signal, so the RNA synthesis will be terminated. Then RNA transcription is finished. Okay, next we go to RNA splicing. Okay, after transcription, the RNA molecule is processed, which is called as RNA splicing. RNA splicing is a process in which a newly made precursor mRNA transcript is transformed into a mature mRNA. During splicing, intron, which is the non-coding region, are removed, and exons, which is the coding region, are joined. So splicing is carried out in a reaction which are catalyzed by spliceosome. Okay, then we have mRNA translation. Okay, in translation, the mature mRNA molecule is decoded in a ribosome, which is outside the nucleus, to produce a specific amino acid chain or polypeptide. The polypeptide later converts into an active protein and perform its function in the cell. Okay, and we can look at the right diagram. From here, we can conclude that the genes from DNA are transcribed into mRNA, then translated into proteins. Okay, for the table, this is the codon. Codon is a sequence of 3 DNA or RNA nucleotides that correspond with a specific amino acid or stop signal during protein synthesis. Okay. Okay, next we look at the mRNA translation process. Okay, translation involves several steps, which are, the first one is initiation. Initiation is when the small subunit of the ribosome binds at the 5' end of the mRNA molecule and move in a 3' direction until it meets a start codon, which is AUG. It then forms a complex with the large unit of ribosome complex and initiation tRNA molecule. Okay, the second one is elongation. The tRNA transfers an amino acid to the tRNA corresponding to the next codon. The ribosome then will translocate to the next mRNA codon to continue the process, which is and it will create an amino acid chain. Okay, and lastly is termination. Translation is terminated when the ribosomal complex reach the stop codon. It's either UAA, UAG, or UGA. Then the ribosome will release the polypeptide. Okay, next is mutation. Okay, the definition of mutation is it is a permanent alteration in DNA sequence, either due to mistakes when the DNA is copied or as the result of environmental factors such as UV radiation, X-rays, or chemical, for example, pesticides or cigarette smoke. Okay, mutation can range in size. They can affect anywhere from a single DNA building block, which is the base pair, to a large segment of a chromosome that includes multiple genes. Okay, the causes of mutation, the first one, it can be due to error made during meiosis and mitosis, which is the replication error. And the second one, it can be due to environmental factors, for example, chemical, radiation, or UV light, which is this can cause the DNA to break down, which make the cell produce a slightly different DNA. Okay, next is the classification of mutation. Okay, mutation can be classified into 3 categories, which are, the first one is genome mutation, chromosome mutation, and gene mutation. Okay, for genome mutation, it is a loss or gain of whole chromosome. For example, monosomy, we can see in monosomy X in Turner syndrome. Next is the example is trisomy. We can see in trisomy 21 in Down syndrome, trisomy 18 in Edwards syndrome, or trisomy 13 in Patau syndrome. Okay, the second one is chromosome mutation. Chromosome mutation is the rearranged genetic material giving rise to visible changes in chromosome structure. For example, of the chromosome mutation is translocation, inversion, and also deletion. Okay, the third one is gene mutation. Gene mutation is the submicroscopic mutation, which is the example is point mutation, frame shift mutation, and trinucleotide repeat mutation. Okay, we will go through deeper for chromosome and gene mutation after this. Okay, next. Okay, this is chromosome mutations. For chromosome mutation, we have four types. We can look at the diagram. The first one is deletion, when a part of a chromosome is left out. Then deletion occurs. Second one is insertion, when a part of chromatic will break off and attach to its sister chromatic, and insertion will occur. The result is the duplication of gene on the same chromosome. And the third one is inversion, when part of a chromosome will break off and reattach backwards. Thus, an inversion will occur. And lastly is the translocation, when part of one chromosome will break off and is added to a different chromosome. Translocation will occur. Okay, next is we take a look at the gene mutation. We have three gene mutation, point mutation, which are point mutation, frame shift mutation, and trinucleotide repeat mutation. Okay, the first one is point mutation. Point mutation is the substitution of a single nucleotide base by a different base. This will result in the replacement of one amino acid by another in the protein product. For point mutation, we have silent, nonsense, and missense. Okay, for silent, it is the substitution occurs in the third position, which do not change the amino acid. The second one is nonsense. The substitution occurs, which result in stop codon. Okay, so this will shorten the translation and most likely lead to a non-functional protein. And the third one is missense, which is the base substitution, which result in the generation of a codon that produce different amino acid, and hence this can lead to a different polypeptide sequence. Okay, for example, missense mutation, it occurs in beta globin chain of hemoglobin, and this can give rise to sickle cell anemia. Okay, the second one is frame shift mutation. The definition of frame shift mutation is the insertion or deletion of one or two base pairs that will alter the reading frame of the DNA strand. For example, we can see in cystic fibrosis, it is caused by deletion of 3 nucleotides from the coding region of a gene, which then resulting in the loss of phenylalanine at the 508th position. Okay, the third one is trinucleotide repeat mutation. Okay, this mutation characterized by amplification of a sequence of 3 nucleotides. Okay, first example we can see in Huntington disease, which is there is amplification, which is repeated CAG codon, then cause addition of extra glutamine protein. Okay, the second one is Fragile X syndrome, which is the repetition of CGG codon in X chromosome. If the repetition occurs less than 55 times, the person may still be normal. If repetition occurs between 56 to 200, it can cause pre-mutation. And then if the repetition occurs more than 200 times, it will cause full mutation. And this will lead to a disease we call as Fragile X syndrome. Okay. And last part is importance of molecular genetics. Okay, Then the importance are, the first one, molecular genetics is essential in giving insight into the heredity, genetic variation, and mutation that will cause disease by specific condition. The second one, it is also important for the diagnosis and management of genetic disorders. And lastly, it is used for the identification of genetic disease carriers. That is, and this is important for genetic counseling in order to help the patient and family members to understand the problem, to understand the risk of recurrence, and we can provide information about genetic testing. Okay, I think that's all.
All right. So around 25 minutes, 20 minutes. Okay. So I think this is the basic one kan. So actually only repetition from your previous apa namanya year 1 or year 2 lectures I think. So I guess all of you already understand these topics. But any question maybe? Anyone? Tak ada. So just apa kita nama quiz lah, refreshing. Yang tak nampak, Omil, open your microphone please, unmute.
Ya, Doktor.
Okay, so dia nak, belum lah, bukan presenter kan. So Omil, is it different between genetic and chromosomal disorder? Tak boleh jawab. Tak boleh jawab pula. Amalin, have to answer. Tadi dengar tak? Tadi clear lah kan? Ada the classification, Mana Amalin? Hilang pula. Amalin, you need to unmute lah because you are the presenter. So, is it different between genetic and chromosomal disorder?
The chromosome disorder is the rearranged genetic material in different changes in chromosome structure. In the alteration in the nucleotide basis.
Okay, I think actually all chromosomal disorder are genetic, yeah, but not all genetic is chromosomal, yeah. Depend on the severity of the mutation. Yeah? If the the gene mutation already affect the chromosome, so it will be chromosomal disorder. It will be inherited. But genetic disorder sometimes only nonsense or point mutation. Yeah? And then it is possible if genetic, it is a new mutation. Yeah? So means the previous previous generation has no disease, but it is a new disease. Yeah? So it is very basic. Yeah? So make it very simple understanding. Jangan fikir yang susah-susah, yang DNA, GGC, GTEC, saya pun tak hafal. Yang basic macam tu, yang clinical punya implication. Ya? So itu you kena faham what is genetic, what is chromosomal disorder. Ya? Another basic question sekarang. Ya. Yang tak nampak ni saya panggil nanti, yang tak ada video. Siti Nuraina. So When double pula suara. Mana si Raina?
Yes, doctor.
Okay, so when you investigate a suspected case of genetic or chromosomal disorder, when do you order chromosomal analysis? When do you order genetic study? Paham tak my question? So sometimes kan we request kan, oh this one syndromic, you have a syndromic baby, okay, baby born with a syndromic phase. So when you order genetic study, when you order chromosomal analysis. Kalau ada syndromic baby itu you order mana satu?
If syndromic, we order chromosomal.
Okay, good ya. So paham ya, mananya yang with the syndromic phase itu usually it is a chromosomal problem. Means the mutation already affect the chromosomal structure or number. Tadi ya ada classification, dia ada problem in genomic mutation, maknanya all the chromosomal number tuh udah berubah. For example trisomy ya down, trisomy 21. Structure, for example translocation of chromosome, sama juga sometimes Down syndrome. So if syndromic phase, we simple the chromosomal analysis, because if the chromosomal anomalies, chromosomal disorder, abnormal number or structure, it will affect the phenotype of the child. Ya. Alright. So if if it is affect the phenotype, dia phase pun dari syndromic phase, kita order simple aja chromosomal analysis. But when you need to ordering genetic study nih, yang lain coba. Pergi yang atas, yang tak ada muka. Abdul Hazim.
I'm not sure, Doctor.
Not sure. So, Anissa, Rasidan. Dah panggil banyak nama.
Yes, Doctor.
Apa? When you need to order genetic study?
Bila patient tu tak ada feature, tak ada dysmorphic feature, tapi
Yes.
kita rasa benda penyakit tu runs in a family.
What disease maknanya itu? Contoh. Yang simple je.
For example, like if thalassemia ataupun cancer.
Okay, if non case lah, dah tahu. Non case yang usually common in newborn lah, in babies tu, in infancy. Kita nak tahu ni sakit apa. Kalau thalassemia, kita tahu patient pun dah pucat, and then dah diagnose thalassemia, we need to check the the alpha mutation, alpha gene tu kan. Kalau yang belum tahu case apa, usually in neonatal lah. Maybe you don't learn about neonatology, but you already learn about some metabolic topic kan. Ada. Saya panggil diabetes, okay, itu pun simple je. Actually we confirm diabetes tu with blood glucose punya investigation kan. Case apa yang kita cek ni abnormal tapi tak tahu case apa, kena confirm with genetic. Kalau yang thalassemia, diabetes kan actually non genetik pun dah confirm lah. easily confirm. Mana nih? Yang senyap nih saya panggil pasti. Ja. Oh, hilang lah. Oh, year 5 lah nih dia diagnosis. Apalah, so usually kalau in case of inborn error of metabolism, so baby itu nampak phenotype oke aja kan, normal, tapi dia ASI, persistent hypoglycemia, sampai or persistent metabolic acidosis. So ada metabolic problem dia. So metabolic only kan, the phenotype okay. Tapi metabolic problem, inborn error metabolism itu kita usually check genetic study. Some of the inborn error is due to the genetic issues ya. Alright. Ini ada jawab G6PD. G6PD pun simple aja kita cek dari G6PD enzyme itu kan. Alright. Dah, anyone? Any comments yang lain?
Doktor, I would like
Yes.
to ask tadi regarding about genetic study tu example dia apa?
Yalah, case-case yang inborn error metabolism tuh. Ada case saya cepuk baru je saya cepuk. Saya tak dengar kan. Ada case baby with persistent hypoglycemia. Baby with persistent metabolic acidosis. Nah, case-case macam tu kita tahu, kita kena tahu lah genetic problem ke only metabolic problem ke. Paham? Paham tak?
Faham.
Alright. So later lah after we test lah baru kita tahu dia ada problem apa. Ada some genetic issues lah. Maybe for example Prader-Willi. Prader-Willi itu kalau later in the childhood dia boleh nampak obvious obesity, tapi kalau in the newborn newborn punya phenotype dia newborn period, dia tak nampak Prader-Willi. Dia face sama aja. Tapi later in childhood dia akan develop extreme obesity. Contoh, Prader-Willi itu genetik ya. So, chromosomal number, chromosomal structure normal. Kalau kita send hunter chromosomal analysis ya normal, bisa. Tapi kalau kita buat genetic study, jumpalah Prader-Willi gene tuh. Lagi satu yang Engelmann, yang common ya. So itu boleh kita as in case of inborn error metabolism in newborn period, atau later pun boleh. Contoh tadi case Prader-Willi itu biasa kita tahu patient dia extreme obesity atau other metabolic issues lah yang not easily confirm ya. Kalau diabetes kan ya confirm dia simple ya, with blood glucose high, simple ya, diabetes. Ada some issues, some disease, some problems that we cannot explain with common investigation. Metabolik usually kan. Metabolik ini kan tak nampak. Metabolic syndrome. Kalau simple obesity ya maybe due to diet nutrition ya kan. Kalau extreme obesity, BMI dia more than 150% more than 1.5 times than normal, so kita kena cari. Contoh macam itu. Barulah later kita baru tahu Prader-Willi with genetic study. Okay. Any other comments, questions? Dah, ini baru basic ya. So yang nanti yang next presenter lah yang more clinical. Kalau tadi yang amalin kan very basic, bosan kan, boring nak dengar. Actually you understand semua, dah paham, tapi pun lupa semua and then tak tahu how to implement. So implementation tu, how to practice ya, yang next presenter. Okay? Alright. Siapa yang bisa next? Okay. Putus-putus. Tak dengar.
Dengar tak?
Okay.
Dengar.
Okay. Okay, I'm Izatun, and today I will be presenting on autosomal disorder. So next slide. So this is my outline. Include the definitions of autosomal disorder, the classification, and autosomal dominant as well as autosomal recessive. Next. Okay, the genetic disorder can be classified into several, such as single gene mutation, chromosome disorder, multifactorial inherited condition, unusual pattern of inheritance, and also teratogen exposure. So my part today will be on single gene mutation. I'll be presenting about the autosomal dominant and autosomal recessive disorder. Next. So what is autosomal disorder? It is disorder of the autosomal chromosome. It can be in any gene in 22 pair of autosomal chromosomes, excluding the sex chromosome. Next. Okay, the autosomal dominant. So by the name itself, dominant, there is a single copy of the gene that is mutated from a parent that is sufficient enough to cause the manifestations of the disease. There is a presence of one abnormal gene on one of the autosome, and the transmissions of this disorder will be vertical pattern transmissions, where the parents will transmit the mutated gene to the child, and each child from an affected parents has 50% of chance of inheriting the abnormal gene. Next. Okay. Although we know that in autosomal dominant transmission is in vertical transmission, but in some patients they may have no history of affected family members, meaning that neither the mom or dad has the mutated gene. So there are several reasons for this. The first one is due to de novo mutation. So de novo meaning new mutations. It is a new mutation that occurred in the DNA of egg and also sperm during the embryonic development. So the example of this de novo mutation will be achondroplasia, or also known as dwarfism. So 80% of the patient of dwarfism will have no family history of having the same disease, and it occur in spontaneous mutations where it associated with increased paternal age more than 35 years old. So why this happened? Because in those who 35 years old men, they will have DNA replication defects occur during spermatogenesis, where later on it will develop one mutation in the sperm. Next. Another reason is due to incomplete penetrance. So what is penetrance? Penetrance is a condition in autosomal disorder in which the patient will manifest the symptoms. So incomplete penetrance, meaning that not all individuals that have the mutated gene will present will have the phenotypic manifestation. So if you see on the diagram, so the mother has the mutated gene, and it will be inherited to the son. Okay, one of the son. But okay, if you see on the second generation, the son. Okay, I call this son as individual 2.1. Okay, so when this individual is is the obligate carrier that show no that show no phenotypic features. So we can say that this individual 2.1 has incomplete penetrance gene. Next. Alright. Often in autosomal dominant disorder show variability in symptoms in which the individual with the same autosomal dominant mutation have different degrees of mutation. So, meaning that some individual may have only mild symptoms, and while some will have more severe manifestations of the disease. Next. Here are the examples of autosomal dominant disorder. Okay, today I will be highlighting on the achondroplasia, Marfan syndrome, neurofibromatosis, and okay, the rest we can just example. Okay, next. For achondroplasia, it is caused by defect in cartilage derived bone, and it is most common skeletal dysplasia in humans that occur in 1 in 25,000 births. So the person will present with bone abnormalities that lead to short stature, microcephaly, flat mid-face, and etc. So the pathogenesis is there is a mutation in FGFR3 genes, where this gene is responsible to produce FGFR3 protein, and this protein will regulate for cell proliferation. So when there is a mutation of this gene, it causes permanently activate the FGFR3 receptor, and it will inhibit the control cell proliferation, and ultimately lead to the impact endochondral bone formation. So what is endochondral bone? So endochondral bone is any bone that develop and replace the cartilage, such as the long bones of the arms, the legs, and also the ribs. So this will cause growth restriction, bone shortening, and also skeletal anomalies. Next. So here are the features of dwarfism, where it mainly affects the long bones. It causes shortened long bones. It can cause rhizomelic or proximal shortening of the limb. It can also cause short metacarpals, short phalanges, or known as brachydactyly, and then short hands, trident hand, where the fingers have the same length with divergence of the ring and the middle finger. And they might also have present with various legs, where there is a medial development of the distal bone of the leg. So it presents like a knee knock. For the flat bone, it is less pronounced. Okay. There will be cradiofacial features, including large head size, frontal bossing, flattened nasal bridge, and also narrow foramen magnum. The patient also can present with spinal lordosis. Yes, spinal lordosis. Next. Okay, the second disease is neurofibromatosis type 1. So it is one of the most common autosomal disorder that affect the NF1 gene, which is a tumor suppressor gene. So the prevalence of this disease is 1 in 3,500 births, and the penetrance is 100%, but the expression is variable. So this is the clinical manifestations of neurofibromatosis, where you can see the presence of Lynch nodules, which is the raised tan-colored hematoma of the iris, However, these nodules will not impact the vision. Secondly is neurofibroma. Neurofibroma, you see that the presence of nodules on the back, on the torso. And then the third is cafe-outlet spots. It is the flat, uniformly hyperpigmented nodules. Usually appear during the first year after birth. Okay, and then the presence of axillary or inguinal freckling. It is just a small patch of light brown that appear on the axillary and inguinal region. So next. So to diagnose neurofibromatosis type 1, so there are several criteria that we have to fit. Two or more of the following diagnostic criteria must be present in order to diagnose this. First is cafe-outlet macules. It must more than 6, and then the presence of skin folding, and also niche nodules more than 2, and optic pathway glioma. It is a tumor that arise around the optic nerve, and also skin neurofibroma more than 2, or plexiform neurofibroma. So plexiform neurofibroma is a larger and also occasional nodular, often found in subcutaneous lesion that may lie along the peripheral nerve trunk. And also distinctive bone abnormality, and also history of neurofibromatosis 1 in family. Next. So the next disease is Marfan syndrome. Okay, Marfan syndrome is an inherited disorder that affects connective tissues in the body. So it's caused by the mutation in FBN1 gene, which affect multiple organ systems such as the cardiac, the ophthalmology, and also the skeletal. So this FBN1, it will provide information for making fibrillin 1. So fibrillin 1 will form the microbills that will give strength and flexibility to the connective tissues and also control the growth. Next. Okay, this is the example pictures of Marfan syndrome, where they were presented with Marfanoid habitus, and they were presented with disproportionately long legs, arms, toes, and fingers. They will have extremely tall and slender build, long and narrow face. They will have high arched neck and also crowded teeth, and it can also affect the sternum, where it will be indented and protruding, and it can also dislocate the lens of the eye in condition what we call as ectopia lentis, and also it can increase the high pressure in the eye. If it affects the lung, it causes cystic changes, and it also can lead to flexible joints, flat feet, and also curved spine. Okay, if you see on the left side of the slide, okay, the first picture, so it is the wrist sign, like this, the wrist sign. Okay, the wrist sign positive when the top of the thumb covers the entire fingernails of the fifth finger when we wrap around the contralateral wrist. Usually we normally we cannot, like kita tak boleh nak fully ni kan, fully wrap around them. So in this Marfan syndrome, they will be the thumb will cover the entire fingernails of the fifth finger. So the bottom is the Steinberg thumb sign, like this. Okay, so the positive test will show distal phalanx of the thumb will protrude beyond the ulnar border of the clenched fist that reflects the longitudinal laxity of the hand and also the long thumb. Okay, next. This is just the summary. You can read later on. Next. So for autosomal recessive is a disorder that is inherited in autorecessive manner. Usually it will manifest when there is both copies of gene pair that have mutation. So there are several of autosomal recessive, which male and female are equally affected, and there will be the trait may have skipped a generation, which is non-penetrance, and it can happen earlier age of the onset, and the affected child has 25% of chance of inheriting the altered gene, and it is complicated. The complicating factor will be consanguinity. I will discuss it later on. Next. Okay, if you see here, both the mom and the dad is the carrier, and it passed down the mutated gene, and 25%, and there will be 25 chance of the child to have this condition. Next. Okay. So consanguinity happen when there is existence of relationship by a common ancestor. So there will be increase in the chance that the both parents will carry a gene that is affected by identical mutation that they inherited. So the risk of to get this autosomal recessive disorder is increased by consanguinity, especially by having first cousin marriage, where there is in 8% of incident, and also although it is rare for consanguinity to happen in Western country, but it is commonly happen in in other part of the world, such as Southern India, Japan, and also Middle East, where the incidence is up to 50%. It's quite high. Next. Here are the example of autosomal recessive disorder, such as cystic fibrosis, phenylketonuria, sickle cell disease, congenital adrenal hyperplasia, fetal ataxia, and the list goes on. So I'll be focusing on the top 3. Next. So cystic fibrosis is a disease of exocrine gland function that involves multiple organ systems that can result in chronic respiratory infection, pancreatic enzyme insufficiency, and other associated complications. So the pathogenesis of this disease will be there is the defect or mutation of the cystic fibrosis transmembrane conductance regulator, or CFTR gene, where this CFTR gene is responsible for as a chloride channel that found in all exocrine tissues. So when there is defect in this gene, there will be effect in the movement of salt and water across the cell membrane. Particularly, there will be decreased secretions of the chloride and also decreased reabsorptions of the water and also sodium. Thus, it cannot maintain a composition of the secretion. Eventually, it will lead to high salt content in the sweat glands secretion, and also it can cause thick, viscous secretion in the lung, pancreas, liver, intestine, and also respiratory tract. In this condition, it is favorable for the bacterial growth. That's why it will eventually lead to infection and also inflammation. Next. Okay, cystic fibrosis, like what I said mentioned before, it can affect multiple organs such as sinus. It can cause sinusitis. If it affects the lung, it can cause sticky mucus buildup, bacterial infection, and widen airway. Noted that it affects the lung, so the lung it can the most causing death is okay, sorry. So next is skin. it can cause the sweat glands produce salty sweat, and liver. It can block the biliary ducts, and it can cause cholelithiasis, cirrhosis, and other form of hepatic dysfunctions. Pancreatitis, intestine, that can cause like chronic diarrhea. Next. The next disease will be phenylketonuria, where it is an inborn error of metabolism in which there is a disorder that affects the amino acid phenylalanine. So it commonly known as phenylalanine hydroxylase deficiency. So this phenylalanine hydroxylase is responsible for the conversions of phenylalanine and also tyrosine. So when there is when phenylalanine cannot be converted to tyrosine, there will be accumulations of phenylalanine in the body fluid in the form of phenylactate and phenylacetate. Next. So the clinical manifestations, usually the newborns or infants, they are asymptomatic prior to initiations of phenylalanine containing foods such as breast milk or standard formula feeding. So, but however, in untreated patients, the hallmark of the disease will be mental retardation, where they will have irreversible intellectual disability, with average IQ less than 50, and also convulsions. They also may have behavior problems, skin rash such as eczema, and also musty body odor due to buildup of phenylalanine in urine and also sweat. Next. The last disease will be sickle cell disease. So sickle cell disease happen when there is a mutations of beta globin chain that will result in sickle hemoglobin or HbS. So in the oxygenated state, the HbS will undergo polymerization, where this hemoglobin will form crystal and eventually cause change in it turn to sickle shape. So for sickle shape hemoglobin, it is less flexible and it is sticky to the vascular endothelium wall, and eventually it can occlude the vessels and cause infarction in the nearby tissues. So this infarction can cause pain, and also it can exacerbate by hypoxia, acidosis, fever, hypothermia, and dehydration. So repeated sickling process can cause irreversible condition where there is a fragmentation of the membrane, and also it can lead to intravascular hemolysis. Next. So this is the pattern of the sickle cell disease, where the both parents carry sickle cell trait, HbA, HbS. So they are heterogeneous. They have heterogeneous gene. So HbA is the normal hemoglobin, while HbS is the sickle cell hemoglobin. So in this disease, the chances for the child to born with sickle cell disease is 25%. There is also 1 in 4 chance that the children are having no traits or normal, and also there is 1 in 2 chance or 50% that the child will get the sickle trait. Okay, I think that's all. This is the summary.
Alright, so question first. Ada double yang my voice ni, ada yang you pakai 2 device ya. Double voice ni. Alright.
I have a question.
So okay, tak ada question?
Doctor,
Yes.
I have a question. So what is the relation between chromosomal and also autosomal disorder? Is it a different entity or just the same?
Oke, siapa tadi? Iza jawab atau Amalin nanti bantu lah, because ini basic lagi balik ke yang tadi. Iza dulu jawab lah, Iza.
I think if chromosomal disorder, it can affect the whole, the structure of the chromosome. For this autosomal dominant, it can I'm not so sure, Doctor. Amalin?
Amalin. Amalin tak ada, hilang, tidur. Mana Amalin? Ada dia, unmute lah Amalin. Mana Amalin? Pergi ke?
Ada.
Yes.
Between chromosomal and autosomal.
Tak sure juga. Kau tak confident jawab je. Kau dah present dah apa? You have already the slide kan. Lah the problem tu apa, maybe you know but don't really understand kan. Don't really know how to implement kan. Okay, so balik ke Faiz. Faiz, so tadi yang disease yang Iza explain tadi. For example, achondroplasia. Apalagi tadi Marfan, and then ada lagi cystic fibrosis, ada lagi thalassemia. So you want to send chromosomal analysis or genetic study?
Genetic study.
Betul lah kan? So maknanya tadi Ijazah dah cakap betul sikit. So kalau the problem tu in the chromosomal number or structure, we send chromosomal analysis. Tapi kalau the problem tu on the it is inherited by the autosome or what we call it sex chromosome link, so we send genetic study, because in these diseases yang macam autosome and then sex link ni disorder, ya chromosomal number and structure will be normal. Tadi actually balik tadi yang my question tadi, kalau you rasa this problem ni, dysmorphic feature ni due to the chromosomal abnormality in the number or structure, kita hantar chromosomal analysis. Tapi kalau ternyata normal, maknanya itu genetic disorder ya. So autosome and sex ini ya kita sebut actually genetic disorder lah, nak bezakan, nak senang lah. Chromosomal disorder or genetic disorder. Kalau chromosomal disorder hantar chromosomal analysis, kalau genetic disorder hantar genetic study ya. So genetic disorder tu boleh yang new mutation macam inborn error metabolism tadi yang saya explain, pada Willy ke apa, ada yang really inherited dah on autosomal or sex link punya link ya. Sex autosomal or X-link disorder. So, yang ini tadi yang Iza is genetic disorder ya. So ada dua semua kan, all this disorder nih yang genetik nih is following apa? Rule apa? What rule? Iza boleh balik lagi atau Amalin? Dia inheritance, mode of inheritance ikut rule apa dia? Rule-nya Mendelian rule ya, Mendelian law ya. So ikut Mendel, dia ada dominan, ada recessive. Autosome itu patutnya tadi Iza ada explain tak tadi? Mendelian law tadi atau Amalin tadi? Tak ada ya? Actually yang better, yang more apa namanya applicable ya belajar pasal Mendelian law tadi. So the autosome X-link ada dominan, ada recessive. Yang Iza explain yang autosome ya, dominan and recessive. So dia ikut Mendelian law, dia inherited. Kalau yang dominan ya 50% chance ya. Kalau yang recessive 25% chance ya. So, contoh sakit ada tadi ya. Dah Faiz, paham, understand?
Okay, faham betul.
Alright. Others maybe comment, questions? Siti Arisha, unmute please. Oh, hilang. Ada. Oke, yang autosome dominan apa? Sebut 3. Tak ada suara dia. Afiq je lah, Afiq. Siti Arisha suara tak ada. Tapi Afiq dah buka video lah. Afiq, apa autosome dominan?
Ya, betul.
Ya apa yang autosome dominan? Mention 3.
Disease,
Yes.
achondroplasia, neurofibromatosis, and also malformation.
Simple kan. Achondroplasia tu dia actually tadi kalau you tunjuk picture dulu senang ya. Achondroplasia short stature, extreme short stature. Dia short long bones. Autosome dominan. So maknanya kalau ada ayah atau mak yang achondroplasia tu most likely itulah 50% of the offspring. Actually ada one TV show kan, kalau you rajin tengok TV itu ada yang family dia memang husband and wife itu memang both achondroplasia. So all offspring ya will be achondroplasia because it is autosome dominant. Contohnya Marfan is extreme tall ya, tall stature. Dia ada contoh dia ada trias organ involvement ya, heart, eye, and musculoskeletal problem ya, macam thumb sign, and then wrist sign ya, thumb sign. Dia tall, itu musculoskeletal. Heart issue dia, Marfan ini dia elongation of aorta dia. Jadi kenapa paham yang genetik ini kan tadi saya dah explain ya. Genetik itu most likely problem tu on the metabolism. Marfani disorder on the metabolism of the protein apa tadi saya lupa. Dystrophin ya. Bukan dystrophin, is Duchenne ya. Ada one gene yang dia problem in the metabolism of the connective tissue kan. So connective tissue disorder of the musculoskeletal ya, tulang jadi panjang, and then on the heart, on the cardiovascular, aorta dia and giant artery dia jadi lembek, elongation, and the eye pun jadi katarak ya, because protein disorder on the lens ya. Itu contoh ya. Achondroplasia pun sama, metabolism problem of the long bone, dia jadi short long bone. And then yang cystic yang autosomal recessive ini contohnya cystic fibrosis, PKU, thalassemia tadi masuk autosomal recessive ya, jangan lupa ya, thalassemia is autosomal recessive. So memang dia problem with the metabolism of the hemoglobin, especially the globin change ya. Terhadap memang kalau ideal thalassemia ini kita hantar lah genetic study, tapi biasa kita dah hantar HP analysis pun dah oke lah, HP beta thalassemia especially. Kalau alpha thalassemia memang kena genetic study itu. Kalau yang beta thalassemia usually from hematologic study pun dah cukup lah. Alright. So, any more comments, questions? Autosome nih? dah starting pening kan, udah banyak diseases yang kena bukan hafal ya, understand. Itulah betul dari Iza, present yang common je. Yang not common janganlah present. Saya pun dah tak paham semua. Okay. Alright. Dah anything? Tak ada? Dah semua senyap dah. Dah penat dah. Baru 2 ni. Ada another 2 presenter kan ya? Okay, go on, next presenter. Alright.
Okay,
Mana ni di slide?
Hello, can everyone hear me?
Yes.
Yes.
Okay, yeah.
Yeah.
Okay, so my name is Fatima Zuna Binti Saini, and I will be continuing our seminar today with the topic of sex-linked disorders. Now, in this part, we'll briefly go through what is sex chromosome and the Mendelian law of inheritance, and then we will go detail into some disorders that are related to the X and Y chromosome. Okay, now as we all know, we have 23 pairs of chromosome, and out of these 23 pairs, one of it is the sex chromosome, which comprises the X and Y chromosome. Now, this X and Y chromosome each plays a role in determining the male and female traits, and they also carry certain characteristics. Okay, and now female, they get both their X chromosome from each of the parents, while male, they get their X chromosome from their mother and their Y chromosome from their father. And these X chromosome are larger than the Y chromosome. Okay, now the sex-linked pattern, it follows the Mendelian law of inheritance, in which it is said, he said that the transmission of inherited traits or diseases is caused by a variation in a single gene in a characteristic pattern. Now, there are 3 laws in this Mendelian law. There are first the law of segregation, in which the 2 alleles for each gene are placed in different gametes. And the second one is the law of independent assortment, in which the inheritance of one gene doesn't affect the inheritance of any other gene. The last one is the law of dominance. This one is the one which Dr. explained before, in which there are, when 2 different alleles are present, only one is dominant and will be expressed, while the other one is recessive and it will be masked. Now, the sex-linked gene, these are genes that are located on either the X or the Y chromosome. And the X chromosome, they contain a lot more genes compared to the Y chromosome due to the fact that they are larger. X-linked inheritance is when there is transmission that involves gene located at the X chromosome. And the diseases that are related to the X chromosome will be affecting both genders because both of them has X chromosome. And if the affected father, they will only transmit the disease to the daughter only, while the affected mother will transmit the disease to both son and daughter. Now, the Y-linked inheritance is when the transmission involves a gene located at the Y chromosome, and this transmission occurs from father to sons only. Now, sex-linked disorders, they are genetic diseases that are caused by a mutation of a single gene located either on the X chromosome or the Y chromosome. Now, for the X-linked disorder, we can divide it into dominant or recessive type. Okay, and these are the diseases that are classified under those categories, and we'll be talking about them. Now, for the X-linked chromosome, since female, they have 2 copies of, they have 2 chromosomes, 2 X chromosomes, so they will have 2 copies of each gene. Hence why they can become either homozygous or heterozygous or a carrier, in which they can either carry two affected gene or only one gene. And since male only has one X chromosome, so they only have one copy of the gene, and hence they are hemizygous. And in X-linked dominant disorder, there won't be any carriers because because they are dominant. So when one mutated dominant gene is present, so it will certainly be manifested. So there won't be any carriers. Carriers are only seen in the recessive disorder. Now for the X-linked dominant disorder, they affect the dominant gene on the said chromosome, and both males and females are affected. But the disorders are more commonly seen in female compared to male. However, they are less severe due to the fact that female has X chromosome inactivation, and this X inactivation I will talk towards the end of this topic. Now for the X-linked dominant, if an affected father marry an unaffected mother, so all his daughters will be affected, and no sons will be carrying the disease. And if an affected mother marries an unaffected father, so about 50% of the children will be affected, and another 50% won't be affected. Okay, so one of the disease under X-linked dominant disorder is the hypophosphatemic rickets. So this disorder is when there is inability of the kidney to reabsorb phosphate. Now we know that phosphate is a vital mineral that is used for bone and teeth formation, and phosphate is largely controlled by the kidney, in which if there is any excess, it will be excreted in the urine, and if there is low amount of it in the blood, so the phosphate will be reabsorbed by the kidney. In this disease, there is too much excretion of the phosphate in the urine that leads to hypophosphatemia. And this disease is due to the mutation of the PHEX gene, in which this gene regulates the fibroblast growth factor 23. And this protein, this fibroblast growth factor, it inhibits the kidney's ability to reabsorb phosphate. So normally the function of the protein will be balanced. However, in this disease, because the gene is mutated, so the resulting protein will either be increased in production or it will be reduced in breakdown, causing it to be staying longer in the blood. In a mildly affected person, they may have hypophosphatemia, but clinically they won't show any sign and symptom. And in severely affected one, we can see that there will be abnormalities in their bone development, in which they can be shorter than their peers, and they have slow growth because there's not enough phosphate for the body to use. So from that, the hallmark or the most prominent sign is the bone abnormalities, which you can see in the picture. It is called the bowed legs or knobbed legs. And in infants, there will be premature fusion of skull bones and so on. So the next disease is the incontinentia pigmenti. Now this is a condition that affects mainly the skin, and it can also affect the other system. And this is due to the mutation of NEMO or IKBKG gene. This gene, it produces the protein called nuclear factor kappa B, in which this protein, it helps to protect cells from apoptosis. So imagine if there is a mutated IKBK gene, hence the nuclear factor kappa B will be mutated also, and then it will lead to cell apoptosis. Incontinentia pigmenti is more commonly seen in females, Why? It is because when a male is affected, usually they will die in early development of the embryo, meaning they won't come into the world. So hence why there is only few males that are born with this disease. And the characteristic of this disorder is that based on the name, there will be swirling pattern of hyperpigmentation that develops after a perinatal skin rash with blistering. So there will be skin abnormalities throughout their life. In child, there will be blistering rash, and then when the rash heals, then develop another kind of skin abnormality called the wart-like skin growth, as you can see in the picture. The top right picture is the characteristic swirling pattern of hyperpigmentation. However, this swirling pattern, as the kid grows and becomes an adult, the swirling pattern will fade and become hypopigmented lines. Hence why when we have patients that we suspect to be having incontinence pigmenty, we can check their mom also for any hypopigmented lines on their arms and legs. And other signs and symptoms include alopecia, small teeth or few teeth, eye abnormalities, lined or pitted nails, and delayed developmental or intellectual disability. Now, to treat this disease, usually it is to address the specific symptoms. The next one is the Rett syndrome, and Rett syndrome is exclusively happen in girls only, in which it is a brain disorder that is due to the mutation in MECP2 gene. This MECP2 gene, it produces MECP2 protein, in which this protein helps in normal functioning of the brain. Hence why in this disease, the normal functioning of the brain is compromised or very, very jeopardized. Okay. In a child with this disease, usually within 6 to 18 months old, okay, they will have a normal life. However, after that, the parents will notice sudden changes in which their child won't talk. They be having they can't even say their name. They can't even like talk. And this is because the syndrome affects the language and communication, learning, coordination, and other brain function. And aside from that, the child may have microcephaly, breathing abnormalities, unusual eye movement, and seizures. And this is coming back to the fact that the brain is affected. And by 2 years old, the girl will adopt a hand washing posture, which they will have ringing or flapping, and means that they have lost all their purposeful hand movement. Now for the last one under X-linked dominant disorder is the Alport syndrome. Now Alport syndrome is a genetic condition characterized by impaired function of the kidney, progressive hearing loss, and eye abnormalities. Now this disease is due to mutation in the CLO4A3, COL4A4, COL4A5 gene, in which this gene, they provide instruction to make a protein, a collagen protein called type 4 collagen. Now in this disease, since the amount of protein produced is reduced, amount of the type 4 collagen protein is reduced, so the patient will present with hematuria because the lack of collagen in the glomeruli will lead to improper excretion of blood and protein materials into the urine. And aside from that, the child can also develop a sensory neural hearing loss because this type 4 collagen, it plays a role in the inner ear, in which it maintains the integrity of the organ of Corti. So we know that organ of Corti it transforms the sound wave into what do we call that? Nerve impulses, so that we can perceive whatever it is that we are hearing. And since the type of collagen also present in the retina, the deficiency of it will lead to misshapen lenses in the eyes. Now moving on to the X-linked recessive disorders. Now I forgot to mention earlier that that X-linked disorders, mostly they are the recessive type instead of the dominant type. Okay, so for the X-linked recessive disorders, if a male is affected, they will definitely manifest the characteristic of those disorders because they only have one X chromosome. While in female, they can either be a carrier or an affected person. So, like in the slide, in order for the female to be affected, they have to receive the recessive gene from both of her parents. And males are commonly affected population in this disorder. Because I said that in the X-linked dominant disorder, females are more affected, but in the X-linked recessive, the males are more affected. So this is for the X-linked recessive. If a father is affected, meaning the father has a mutated gene on the X chromosome, all the daughters will be carriers because the mother is unaffected, and no sons will be affected. However, if the mother is a carrier and the father has no mutated gene, 50% of the son will be affected and 50% of the daughter will be a carrier. So the first one is the most common one, which is the Duchenne muscular dystrophy. Now, it is a genetic disorder that is characterized by the progressive muscle degeneration and consequently resulting to weakness, and it commonly affects the skeletal muscles and also the cardiac muscles. Now, in this disease, the mutated gene is the DMD gene, in which this DMD gene, it helps to produce dystrophin. Dystrophin is a protein that helps to stabilize and protect the muscle fibers. So if the dystrophin cannot be produced in a normal amount that is sufficient for the for the muscle, so hence why there will be manifestation like in this disease. So the symptoms include delayed motor skills, in which the patient may have difficulty in standing, or they may have delayed walking. So this can be your top differential diagnosis. And aside from that, since this disease, the Duchenne muscular dystrophy, it has a proximal weakness. So hence why there is enlargement of the cup, because the body needs to compensate for the proximal muscle weakness. Aside from that, the patient can also have waddling gait. Next one is hemophilia. Now, hemophilia, as I'm sure you guys are very well aware of it, it is a bleeding disorder that slows down the blood clotting process. Now, hemophilia, it can be divided into hemophilia A or hemophilia B, in which in hemophilia A, there will be factor 8 deficiency, while hemophilia B consists of factor 9 deficiency. Now, both of these deficiency are contributed to their respective gene. Like for the hemophilia A, the F8 gene is mutated, and for hemophilia B, the F9 gene is affected. Next one is color vision deficiency or color blindness. The most common color blindness is the red-green color vision. So if we normal person, we can see green, we can see red. However, in a person with color blindness, yeah, that is how they will see objects, green objects or red objects. They will appear like gray and murky. So they have trouble in distinguishing between some shades of red, yellow, and green. The mutation occurs at the gene called OPN1LW and the rest. So the next one is the common one. It is the G6PD deficiency. Now, it occurs almost exclusively in males. However, if we see a female child with G6PD, so it can either be, so we have to really confirm with the lab whether that is true, or it can be that both parents are the carrier of G6PD deficiency. Okay. Okay, G6PD is a genetic disorder that mainly affects the red blood cells, in which the defect in G6PD will lead to hemolysis and hemolytic anemia. So the patient comes with a dark urine, pale, shortness of breath, all those symptoms related to hemolytic anemia. And usually these conditions are triggered by infection, drugs such as antibiotics such as sulfonamide, and antimalarial drugs. And the most famous triggers are the flavavins. Now, G6PD is a spectrum. Sometimes a small amount of trigger can cause the effect. And in some patients, a large amount of trigger is needed to cause the effect. Okay. So for the X inactivation, this only happens in female because we have 2 chromosomes. There has been studies that say that 1 chromosome is sufficient to cater to our body's need, meaning the gene activity is enough. So since we female have 2 chromosomes, 2 X chromosomes, hence why our body inactivate one of it. What I mean is that in each cell, other than the egg cell, only 1 X chromosome will be functioning. It can be either the maternal X chromosome or the paternal X chromosome. And how the body chose which chromosome, which like father or mother X chromosome to inactivate it is random. So one cell, it will inactivate the maternal X chromosome, but in the other cell, it will inactivate the paternal chromosome. So both chromosomes have equal chances to be inactivated. And this process is to ensure that the females have 1 functional copy of X chromosome. So moving to the last one, it is the Y-linked disorders. Now, these disorders, it is extremely rare because most Y-linked genes, they are related to, because most of these disorders are infertility. So they can't reproduce more and transmit the disease. Okay. And it is a male-to-male transmission only. And aside from infertility, which is the common one, other Y-linked disorders include retinitis pigmentosa and auricular hypertrichosis. Now, retinitis pigmentosa is when there is a breakdown and loss of cell in the retina, leading of loss of vision. And usually, there will be progressive loss of visual field starting from the periphery. So the patient will have a tunneling vision. So they will have difficulty in performing tasks. Next one is auricular hypertrichosis. Now this is excessive hair growth within or on the ear. They don't really possess harmful effect to the body aside from physically. Now the differences between sex-linked inheritance, which I have gone through. So in X-linked dominant and X-linked recessive, there is no transmission from father to son. However, in Y-linked transmission, there is a Y-linked disorder, there is a transmission from father to son. And for carriers, it is only seen in the X-linked recessive. And the rest you can read because I have explained this before. So this is the differences between the autosomal disorder and sex-linked disorder. So what differs between my part and Iza's part is that in Iza's part, the autosomal disorder, it appears in both sexes equally because it affects the autosome. While in my in sex-linked disorders, it appears more common in males because like they have one X chromosome, which is the related to the X-linked recessive disorders, and yeah. So in autosomal disorder, the affected alleles can be inherited from either parents, while in sex-linked disorders, the different genders of children receive from either mother or father. And these are my references. Is there any question?
Okay, any comment? Tak ada. So tadi saya salah ya. Yang penilaian rule ini only for X-link, not for all any mode of inheritance ya. So sebenarnya autosome ni we just expect ya autosome dominan will be 50% chances, kalau recessive 25% chances. Kalau yang X-link ni ikut Mendelian dari rule ya. So, ada dominan, ada recessive. So most likely yang X-link dominan tu dia letal. So that's why all male usually will not survive. So you will see common case of abortion kan in pregnancy tu. One of the reason of abortion tu yang inilah, yang lethal diseases. So itulah memang dah kuasa Allah macam tu ya. So dia tak nak, Allah ni tak nak bagi severe situation that we cannot afford kan. So kalau dah lethal disease ni memang tak survive lah, memang abortion. Okay, kalau female maybe boleh survive, but with with some manifestation ya. Okay, yang most common is X-linked recessive, which most likely affect male only ya, but ada some inactivation that female can manifest ya. Only very rare lah. Dan yang Y-link pun dia bukan lethal tapi infertile ya, so that's why yang Y-link pun juga very rare. Okay. So nak 3S nih tadi yang Nuraina dengan Nuraina dah ya, yang ini dengan Airisah dah kan, yang belum nih Sarah, Sarafina dengan Sarifah mana? Sarafina dah open video.
Yes, Doctor.
Oke, Sarah dulu. Contoh yang X-link dominan apa?
Contoh X-linked dominant. For example, incontinentia pigmenti, Rett syndrome, Alport syndrome.
Okay. Incontinentia pigmenti jarang jumpa. Yang biasa kita discuss tu Rett and Alport lah. Rett tu neurological disorder. Yang Alport tu kidney lah, tapi ada eye dengan hearing disorder juga. Okay.
Yes.
Alright, and then Sarafina tadi. Sarafina mana?
Ya, Doktor.
Okay, yang X-linked recessive apa?
G6PD, hemophilia, color blindness.
Okay, ya. So paham ya. Ada lagi yang most common apa tadi? Yang first?
DMD.
Duchenne ya. Semua itu adalah X-linked recessive. Dia di metabolism of the protein dia affected. Dan kalau yang dystrophin gene tadi, so jadi muscle weakness ya. So weak tapi dia hypertrophy ya. Dia hypertrophy of the calf. Tapi karena weakness jadi word linkage, lama-lama jadi gower sign. Kalau udah affect di heart jadi myocarditis lah, or cardiomyopathy. Okay. Dan Yang color blindness ya affect the protein of the on the eye or on the receptor or on the brain. Kalau color blindness tuh.
On the receptor.
Receptor yang on the eye. Okay. Hemophilia, protein of the coagulation factors ya. A, B, and C or non A non B sekarang hemophilia nih. Kalau hemophilia ada berapa type, Sarafina?
2 type.
Apa?
Hemophilia A, hemophilia B.
Sudah ada third, yang non A non B pun boleh. Dia yang A kan faktor 8, yang B faktor 9, ada yang faktor 11 tuh, non A non B. Atau ada yang sebuah hemophilia C juga, yang very rare ya. Oke, last kali Sarifah, mana Sarifah?
Yes, Doctor.
Oke, Sarifah for Wailing apa?
Example ada male infertility, retinitis pigmentosa, and also auricular hypertrichosis.
Oke. Hypertrichosis nampak ya, telinga dia, rambut dia panjang. Retinitis pigmentosa nih, dia simptom dia apa?
As for the symptom, night blindness.
Oke, night blindness ya. So simple ya, night blind. Putus-putus pula Sarifah nih. Oke, night blindness nih.
Night blindness,
Kalau ada
progressive loss of
kalau ada case night blindness,
and also
Oke, tak apa. So kalau ada night blindness, so do you think directly on retinitis pigmentosa or any other diseases?
in vitamin A deficiency.
So jangan lupa ya, actually more common is A vitaminosis, deficiency of vitamin A. Alright. Okay, any more questions? Others? Alright, so last ya, lagi satu. Dah cepat dah panas tengah hari. Who is the last presenter? Mana?
Me,
Faisal. Okay, alright.
Okay. So I will now present about the chromosome disorder. I hope all of you can stay focused. Okay, next. So what is chromosomal disorder? It is an error that occurs in meiosis during the production of gametes, in which it will lead to the abnormality at the number or structure of the chromosome. For the number disorder, it can be trisomy, which is Down syndrome, Edward or Patau, or monosomy, which is Turner syndrome. For structure, it can be deletion, translocation, or inversion. However, in this topic, I will be explaining all of the disease. Next. So for the Down syndrome, the name is trisomy 21. Next. So Down syndrome is when patient has 3 copies of 21 chromosome in every cell. So Down syndrome is most common abnormality of chromosomal number. It occurs in 1 of every 1,000 birth. However, it is highly related with the maternal age. As you can see the table below, if maternal age increase, the risk for Down syndrome also will be increased. Next. So how can the patient get Down syndrome? There is 3 ways that patient can get Down syndrome. The first and the most common case is by non-disjunction, which is 64%. And the second one is translocation, 5%. And last one is mosaicism. So for the non-disjunction, most cases result from an error at meiosis at the meiosis 1 or meiosis 2, in which the chromosome 21 pair fails to separate and one gamete has two chromosome 21 and one has none. So if there is fertilization occur of the gamete, two chromosome 31 give rise to a zygote with trisomy 21. And for the translocation, there will be extra chromosome 21 is joined onto another chromosome. For example, chromosome 14, which is most common. It is called Robertsonian translocation. It may be present in phenotypically normal carrier with 45 chromosome, or in someone with Down syndrome and a set of 46 chromosome but with 3 copies of chromosome 21. As for the mosaicism, it is rare, and mosaicism is mean that when the patient has two or more genetically different set of cells. For in this case, for Down syndrome, some of the cells are normal and some have trisomy 21. So in patient with mosaic Down syndrome, they have milder sign and symptom compared to the other Down syndrome. Okay, next. So this is the illustration of the non-disjunction and Robertsonian translocation. As you can see, we focus to the left figure first, which is non-disjunction. You can see that basically one parent creates two normal gametes and the other parents creates one gamete with two copies and the other one is simply isn't viable with no copy of trisomy 21. With no chromosome 21. And then when fertilized, the patient can have trisomy 21. And then we focus on right figure, which is Robertsonian translocation. Here we have one parent with two normal gametes with one copy of chromosome 21, which are the red color, and one copy of chromosome 14, which is blue color. And the other parent is a translocation carrier. As you can see, the carrier will make two possible gametes, as you can see in the figure. And the left one being a normal gamete, and the other 5 are variations. Now, when these two parents make an offspring, it is possible that they make a normal offspring, which is the left one. As you can see, there will be 6 possible outcome of the offspring, and 3 of them are not viable, and 3 of them is viable. And the viable offspring, one can be normal, one can be the carrier, the same as the carrier parents, and one can have the Down syndrome. Okay, next. So these are the clinical feature patient with Down syndrome. First, we go to the typical craniofacial appearance, where we can see patient will have round face and flat nasal bridge, and then up-slanted palpable features, epicytic fold, bush-filled spot in iris, small mouth and protruding tongue, small ears, and flat occiput and third fontanelle. Other anomaly include short neck, single palmar crease, as you can see in the figure, in the diagram, and then incurve and short fifth finger, also known as clinodactyly, and wide sandal gap between the first and second toes, as shown in the picture. Next. This is another picture. We show the symptom sign of patient with Down syndrome. You can see the bush-filled spot on iris, and then sandal gap and small hypoplastic ears. Next. Down syndrome also can affect many other system or organ, such as about 50% of patients with Down syndrome will have heart disease. So the common congenital heart defect that patient can have is AVSD. Other less common is VSD, ASD, TOF or PDA. For the GIT, it occurs on 12% of patients with Down syndrome. The common one is duodenal atresia. Other is H-bone disease, pyloric stenosis, and anorectal malformations. It can also affect the vision, with the infant or children can present with congenital cataract or glaucoma. It also can cause hypothyroidism and also hypogonadism. Next. Patient with Down syndrome also can have development and growth problem such as delay development, learning difficulty, and also small stature. It can also get a infection on the respiratory and also upper airway obstruction, and can have congenital hip dislocation and atlantoaxial instability. And then for hematological, it can have tension abdomen myopoiesis and also leukemia, especially AML and ALL, which is relative risk is 15 to 20 times than the normal persons. Also in puberty, boy with usually infertile. For the girl, usually they have slightly delayed puberty. So in adult life, patient with Down syndrome have increased risk of dementia and also Alzheimer and shorter life expectancy. Next. This another figure and also how to memorize the sign system of Down syndrome. You can read it later. Next. So how to diagnose patient with Down syndrome? We can diagnose it antenatally and also newborn. So antenatally, we can do ultrasound. In ultrasound, what we expect is patient with Down syndrome will have nuchal fold thickness more than 3 millimeter. And so we need to look sign for double bubble sign, which is indicate the duodenal atresia, which is the commonest DIT presentation of fetal with Down syndrome. And then we can also do amniocentesis, which is can do it at the 6th or 5th acting weeks of gestation. Then we do calotyping, which is chromosomal analysis. And also we can do FISH, which is the short term for fluorescent in situ hybridization. And then we can also do chorionic villus sampling and do the karyotyping after that. The karyotyping is the diagnosis, is a confirmation test for patient with Down syndrome. Other than that, we can also do a triple assessment, which include the blood marker, include the alpha fetoprotein, estradiol, and human chorionic gonadotropin. So in patient with Down syndrome, we expect that there will be low level of AFP and estradiol and high level of HCG. This is done by taking the blood of the pregnant mother with the suspected fetal of Down syndrome. And also for the newborn, we can do blood sampling and then we do calotyping and also FISH. Okay, next. This is the illustration of, you can see the nuchal thickness is more than 3 millimeter by ultrasound. And so we can see the duodenal atresia, which is the double bubble sign. The first bubble is in the stomach, and the second bubble is after the obstruction of the duodenum. Next. This is FISH fluorescent in situ hybridization analysis, in which we use a fluorescent probe which are complementary to the part of chromosome. And in here we can see that there is three red color which indicates that trisomy 21. Okay, next. And then this is the procedure that can be done antenatally, amniocentesis and also chorionic villi sampling. Next. So the management is mainly as listed here. The first is communicating the diagnosis is preferably handled in private by a senior medical officer or specialist, which is familiar with the natural history and genetic aspect. Other than that, it is important to do a careful examination to look associated complication. For example, the investigation it can be done is echocardiogram by 2 weeks or 6 weeks. Other than that, chromosomal analysis and also T4, TSH at birth or by 1 to 2 weeks of life because patient can also have hypothyroidism. So early intervention program should begin at diagnosis if health condition permits. And then it's important to assess the need of family, contact with local parent support group should be provided. So and then last we do have surveillance and monitoring. Next. So this is the table that shows the series of follow-up for monitoring the health problems or complication that might arise with patient with Down syndrome, as you can see here. Okay, next. Now we will move on to the Edwards syndrome. Next. Edwards syndrome is a disorder in which the patient have 3 copies of chromosome 18. This is the second most common trisomy after Down syndrome. It occurs 1 in 7,500 live birth and 80% are female. So the nomenclature for Edwards syndrome is 47XX plus 18 or 47XY plus 18. As low survival rate, more than 95% spontaneously aborted in first trimester. So the clinical features include small and premature appearance, hypertonia, prominent occiput, micrognathia, which is small mandibula, short sternum and clenched hands, as you can see in the picture. Other clinical features are low birth weight, small and mouth and chin, short sternum, vocal bottom feet, and cardiac and renal malformations. Next. So for the diagnosis of Edwards syndrome, many affected fetus are detected by ultrasound scan during the second trimester of pregnancy, and the diagnosis can be confirmed antenatally by amniocentesis and also chromosomal analysis. It also can be diagnosed on non-invasive and prenatal testing. So how the patient with Edwards syndrome can get the trisomy chromosome 18 is the same as the Down syndrome, which is by nose disjunction, which is a common case, and followed by Robertsonian translocation and mosaicism. Okay, next. So now we move on to the Patau syndrome, trisomy 13. This is the third most common trisomy. It occurs in 1 in 5,000 live birth. So this is due to the chromosomal disorder where a person inherit an extra copy of chromosome 13. It is the same with other trisomy. How can patient get Patau syndrome is by the common case nodus junction followed by Robertson-Liam translocation and also mosaicism. So usually patients with Patov syndrome are fatal in the first year of life. Only 8.6% survive beyond the first birthday, 1 year of life. So they can have severe intellectual disability and physical abnormality, can have numerous malformation. Other clinical features include structural defect of brain, scalp defect, small eyes, microptomia, and other eye defect such as cyclopia, and then left cleft lip and palate, polydactyly, more fingers, cardiac and renal malformations. Next. So as I mentioned, Patov syndrome is much less common compared to Down syndrome, but it can be presented with more severe presentation. Most baby dies before birth due to the many problems with the malformations. So as you can see, they can have cleft lip and palate, and dysplastic or malformed ears, polydactyly, extra finger, and undecided or abnormal testes. They can also have severe presentations such as cyclopia. As you can see, they have one orbit only, single orbit. And so they can have cutis aplasia, which means cutis is skin and aplasia is failure to develop. In this, we can see that the below right figure, we can see that the patient, the skin on the scalp fail to develop, and they can also have don't have nose. The nose might be missing and replaced by proboscis. As you can see, in the figure, like a trunk of an elephant. Okay, next. So this is the table we show the manifestation of trisomy 13 and also trisomy 18. I want to highlight is that for trisomy 13, only 5% live after 6 months, and for trisomy 18, only 5% live after 1 year. Okay, next. And then we move on to the Klinefelter syndrome. Next. So Klinefelter syndrome is the most common sex chromosome abnormality affecting about 1 in every 500 male birth. So it is the most common cause of hypogonadism and infertility in men. It is caused by the presence of at least one extra chromosome. Rarely, in severe form of Klinefelter syndrome occur when there are at least 2 or more extra X chromosomes, such as 48 XXXY or 49 XXXXY. So before puberty, boys with Klinefelter syndromes are phenotypically indistinguishable. So which means that the symptom of Klinefelter syndrome most apparent around the time of puberty, because that is when the low level of testosterone are most evident. So diagnosis often made when the boy is at 15 or 16 years old. As you can see in the picture at the right, you can see that the long chromosome, which is the X, have extra copy. Okay, next. So this is the clinical feature of patient with Klinefelter syndrome. As you can see, infertility is the most common presentation. And patient can also present with hypogonadism with small testes. Pubertal can have a delayed pubertal development only if they been given testosterone therapy. Other than that, they can develop gynecomastia, tissue short breast, and then it can presented with tall stature and also will have intelligence problem usually in the normal the intelligence usually in the normal range, but some have educational and psychological problems. Next. So patient with Klinefelter syndrome typically have small testes that produce reduced amount of testosterone, which this is by primary testicular insufficiency. And then the testes will fail to produce viable sperm, and this will result in failure to develop later secondary sexual characteristics such as facial hair, deepening of the voice, and also libido. Later in life, patient also can have osteopenia and osteoporosis. So most men with Klinefelter syndrome are infertile because they produce few viable sperm. Next. So the treatment for the patient with Klinefelter syndrome is first by giving testosterone therapy in order to correct the testosterone deficiency, to provide appropriate visualization and to improve psychosocial status. And also for the treatment for infertility by referring to a reproductive or infertility specialist to help them with various reproductive options. Next. Next, we'll move on to the Turner syndrome or monosomy 45X. So, Turner syndrome occurred in 1 in 3,200 live-born female. So, 99% of embryo with 45X are spontaneously aborted. It is not associated with advanced mother age. Rather, it is believed that the 45X karyotype result from loss of X or Y chromosome after conception. As you can see in the figure, there's only one sex chromosome, which is the X. Next. So these are the clinical features of patients with Turner syndrome. As you can see, they can have short stature, low hairline, widely spaced nipple, delayed puberty. They can also have congenital heart defect, particularly coarctation of aorta. Other than they can have elbow deformity, poor breast development, renal anomaly, pigmented mole, and recurrent otitis media, and normal intellectual function in most cases. Next. So the investigation it can be done prenatally by amniocentesis and also chorionic venous sampling. Karyotyping can be done either by either way, amniocentesis or CVS. If the ultrasound of fetus reveal a nuchal cystic hygroma, caution kidney, left-sided cardiac anomaly or non-immune fetal hydrops. And then it is highly subjective if there is elevation of SCG, FFP, and also estradiol. So standard 30 cell karyotype analysis is required. Diagnosis is confirmed by the presence of a 45X chromosome. Next. So in many cases, a girl with Turner syndrome escape detection during the newborn, yet because phenotypic features are subtle. So about 33% are diagnosed in the newborn because of the presence of heart disease and physical feature, and 33% are diagnosed in the childhood because during a workup or for short stature, and 33% during adolescence when they fail to develop secondary sexual characteristics. So the management will be to do heart monitoring, cardiac evaluations, BP measurement, echo at baseline, and every year is recommended. Other than that, by giving growth hormone therapy for those who have short stature, and also give estrogen replacement for development of secondary sexual characteristic at the time of puberty. However, the infertility persists. Next. These are the deletions, the Chay-Duchat syndrome. Next. Okay. So Chay-Duchat syndrome is a rare genetic disorder in which portion of chromosome 5, which is the short arm, is deleted or missing. Hence, the name 5p minus or monosomy 5p. Other than that, other name is 5p deletion syndrome. The P is referred to the short arm. So the name is a French term that refers to the characteristic of high pitch cry of the affected baby, which sounds like the cry of a cat. Hope you can hear this. Okay. Can you hear the baby crying?
Yes.
Okay. So it is just like cat meow. So most cases arise de novo. So the deletion is usually in the chromosome 5 inherited from the father. Next. So these are the clinical feature of Chay-Duchat syndrome, which is the common one is cat-like cry during early infancy due to tracheal hypoplasia, and then low birth weight and postnatal failure to thrive. And then hypotonia, development in disability, microcephaly, and craniofacial dysmorphism, which include ocular hypertelorism, epicanthal fall, downward obliquity of the palpebral feature, and a low set malformed ears. Other than that, patient can manifest as cleft or lip, cleft of the lip and palates, and congenital heart disease and other malformation may be seen. Things to note that the clinical severity of the Chay-Duchat syndrome depends on the size of the chromosomal deletion. So the larger the deletions, the severe the expressions of the clinical features. Next. So the diagnosis of Chay-Duchat syndrome is by suspect by the clinical feature, which is the main one in high pitch cry by the baby. And then we can do karyotyping if we can see there is a missing short arm on the chromosome 5. If the clinical suspicion is high but karyotyping is negative, we can do fish. And in fish we can see in normal cell we can see there is two color of the chromosome. In 5p deletion, there will only one color appear under when we see under the microscope, under fluorescent microscope. Okay, next. Next, the last one is Prader-Willi syndrome. Next. Wait. So, in Prader-Willi syndrome, some 60 to 70% of individuals with Prader-Willi syndrome have a small deletion of chromosomal 15. This is deletion de novo because the patient have a normal chromosome received from the parents. However, the deletion occurred as a new mutation in the child, which is mainly if there is deletion, the deletion is if the deletion of part of chromosome 15 is from the father, from the chromosome received from the father, it is called Prader-Willi syndrome. It is the deletion is on the chromosome 15 inherited from the mother. It is Angelman syndrome, which is opposite of the Prader-Willi syndrome. So in other case, the individual without deletion, 20% have uniparental disomy of chromosome 15, which means that this patient receive two chromosome 15 from one of the parents. Other parents they receive none. So in Prader-Willi syndrome, the patient have the patient gets two chromosome 15 from the mother and none from the father, which is opposite to with the Angelman syndrome. Uh, they get two chromosome 15 for the father and none from the mother. So it is basically the missing of the chromosome 15 from the father. So it occurs in approximately 1 in 10,000 infants. So it is characterized by hypotonia of prenatal onset and then postnatal growth delay, a characteristic appearance including almond-shaped eyes and also small hands and feet, and then developmental disability, hypogonadotropic hypogonadism, and obesity after infancy, just like Dr. Taufiq has mentioned before. So early in life, the affected infant are so hypotonic that they cannot consume enough calorie to maintain their weight. And then it is Nasogastric feeding is invariably necessary and failure to thrive is common. So later in life, after the few years of life, muscle tone improve and children can develop a voracious appetite. With this condition, patient will have hyperphagia, which is excessive eating, that can lead to life-threatening obesity after infancy. Okay, next. So these are the list clinical feature for the Prader-Willi syndrome. So it manifest in the fetal, such as reduced fetal movement or abnormal life, and then lethargy, hypotonia, poor feeding due to the poor muscle tone affecting the sucking reflex, and then hypogonadism. So in the childhood, as you can see, patient can have hyperphagia at the age of 2 until 8 years old, which proceed to adulthood opposed to infancy poor feeding. Because of this, the patient can have excessive weight gain and also can have obesity. Other than that, the patient might have delayed puberty, short stature, extreme flexibility, and in adulthood, the patient can have infertility, hypogonadism. And then the physical appearance in patient with Prader-Willi syndrome, we can see there is a prominent nasal bridge, small hand, feet with finger tapering, soft skin, easily bruised, excessive fat, especially central obesity, height, narrow forehead, thin upper lip, almond-shaped eyes, light skin and hair, and lack of complete sexual maturity. Other than that, patient can also have neurocognitive problem such as learning and attention difficulties and also speech poorer than comprehension due to hypernasality, extreme and insatiable appetite with morbid obesity and hyper intolerance, unaware of appendicitis, cholestasis until later. Next. So the diagnosis of Prader-Willi syndrome is initially based on clinical diagnosis, and then currently via genetic testing by do DNA-based methylation testing to detect the absence of the paternal region on chromosome 15. So the management for these patients, there is no cure. However, we can treat the symptom by by doing physiotherapy, speech and occupational therapy. Other than that, the recombinant growth hormone injection with the short stature. Other than that, we give positive airway pressure machine, which is due to the obstructive sleep apnea secondary to obesity. Other than that, we do the weight reduction for the patient with obesity, if possible.
Faisal. Sorry, I cut sikit because you are too long lah. More than 30 minutes, semua dah penat kok. Yang tak penting-penting
Okay.
macam manajemen tak payah lah.
Okay,
Habis
dah
dah. Itu last slide dah. Panjang
Dah.
sangat. Because maybe we need to focus on di genetic and chromosomal only lah. Yang lain-lain tak payah, manajemen tu tak payah. Nanti discuss in other topics, other session ya. Okay. So, questions, comments tak ada? Semua dah senyap dah. Yes, Sarifah.
So in Down syndrome, for the non-disjunction disorder, the mother have a phenotype of Down syndrome, but in translocation, can mother or father without phenotype of Down syndrome cause translocation, cause produce a baby with Down syndrome with translocation? Faham tak?
Okay, Faisal, you answer first.
Can you repeat the question? Sekat-sekat.
Can I say it in Malay?
Okay.
Macam ni. Kan kalau non-disjunctional tu kan mak ayah dia okey je, tak ada Down syndrome pun tak apa. Tapi kalau dalam translocation tu kan, mak ayah dia tu memang kena ada Down syndrome ke mak ayah yang normal pun boleh produce baby yang ada Down syndrome through translocation.
From what I know, for the non-disjunction, the parents might don't have Down syndrome, but the non-disjunction occurred during the meiosis. So the parents will not have problems. So when you do karyotyping in the parents, you will not find anything because the non-disjunction occurred in the meiosis. So the karyotyping will be positive if you do to the patient or the child with Down syndrome only. And then for the Robertsonian translocation, one of them need to be the carrier. One might be normal and one the translocation carrier. So if you do, from what I have read, if you can, but the carrier will not might live as a normal. No, they don't express the manifestation of Down syndrome because they are just the carrier. But if you do the karyotyping in one of the parents, there you might found the Robertsonian translocation on one of the parent, which is the carrier.
Okay, the answer actually the translocation tu happen on the gamete, on the kalau mak on the ovum, kalau ayah on the sperm, So actually most likely the parents can be normal actually the phenotype, tapi kan all the ovum abnormal kalau abnormal kalau mak kalau father all sperm has the translocation tu. Alright. Dah paham Sarifah? Okay.
So doktor, maksudnya semua sibling dia akan ada Down syndrome lah.
Faisal?
Tak semua akan ada.
Translocation.
Oh, Translocation, yeah. For translocation, like I mentioned, there is a 6 possibility of the offspring. 3 of them are not viable, and 3 of them are viable, and one of the viable is can be normal person, normal children. One of them can be a carrier, and one of them with Down syndrome.
Tapi itu apa very complicated sangat lah. Simple actually kalau translocation all sperm or all ovum abnormal, ya all offspring semua anak ya Down syndrome lah, simple aja. Kalau explain macam tu memang lah ada possibility yang non viable, possibility yang carrier, tapi itu very rare. Yang simple aja. Macam hemofilia pun in male ya senang lah kan. Memang ada possibility female, tapi kan very rare di lionization. Kalau Down syndrome translocation nih, nah, ya semua lah, semua anak, simple aja. Jangan pikir yang advance yang complicated dulu, you pikir yang simple dulu. Kalau all sperm dia abnormal, mananya anak dia Down syndrome ke tak? Ya all will be Down syndrome kan, simple.
Down syndrome.
Alright, Syarifah paham?
Paham Dr.
Alright. Okay, welcome. So tadi yang my mistake tadi yang Prader-Willi ya, I thought it is only genetic, but it is deletion of chromosome 15 ya. So we send karyotyping lah for Prader-Willi. So not necessarily genetic studies, with only karyotyping we can find. Okay ya. So last few lah yang tadi. Afik tadi udah panggil belum? Afik, dah.
Yeah.
Ada.
Yeah.
Alright, so kalau yang Faisal punya case ini, you hantar apa? You hantar karyotyping atau hantar genetic study?
Karyotyping.
Okay, good. Fatnin.
Yes, okay.
Alright. Tanya apa dah? Dah penat dah sebab last dah. Yang tadi, yang Edward syndrome tadi. Problem dia kat mana?
Trisomy 18.
Faisal tadi saya pun terlupa kalau Edward dia survive berapa lama? Saya lupa tadi. Kalau yang Pato not less than 1 year kan. Yang Edward dia survive berapa lama? 1 year juga sama ya dengan Pato ya?
6 months.
Yang apa?
Trisomy 13 6 months.
Oh, earlier ya. Yang Pato 1 year. Bukan yang Pato lagi fatal ke?
Pato
itulah ya kalau tengok current kalau tengok itu kan isinya Edward umur 6 bulan tak sampai 6 bulan dia pass away, tapi actually ya kalau you baca current experience in Japan tuh ada yang Edward dia survive. I don't know how Japanese people to do all all or none maximum capacity of the medication dia buat, ventilate semua, ECMO semua buat, you survive lah patient tu sampai 2 years and above, tapi ya vegetatif lah, do nothing. So that's why kan survival rate tu actually ya 6 bulan, tapi kalau nak buat semua pastikan organ dia function, heart, lung semua function, actually boleh more than 6 years, 6 months, tapi for what reason kan? If we support the patient with all machine, tapi the patient cannot do nothing. They can do nothing, cannot do anything. Right? Okay. Apa nih yang tak nampak lagi nih? Maya, last sekali lah nih kok. Ada Maya?
Yes, Doctor.
Maya kalau apa? Klinefelter you hantar apa? Patient Klinefelter syndrome.
We send for karyotyping.
Sama ya, simple karyotyping ya. Oke, last kali Hafiz. Tadi Kraiducha tadi apa sindrom? Hafiz mana Hafiz? Kraiducha tadi? Okay, how do you apa diagnosis itu apa ya? Tadi pakai karyotyping jumpa apa? Ingat tak?
Chromosome 5. Oke,
deletion ya. Oh, nah, nak balik Faisal nih apa? Dijo syndrome you baca tak? Dijo syndrome.
Tak.
Dijo syndrome tu dia deletion chromosome 22. Nah, itu actually ya rare tapi apa? Ya we have the patience nah. Nanti yang belum present ini baca Dijo syndrome sikit ya. Ya TOF. And then last kali tanya balik ke siapa nih? Tadi yang autosome tadi siapa? Iza ya? Yang autosome tadi Iza.
Yes, sir.
Iza.
Noonan syndrome you baca tak?
Tak. Just on
Noonan
surface.
syndrome itu dia another autosome dominan ya. So, nanti yang homework for all tuh baca Noonan dengan Dijos. Itulah yang common cardiac tuh. Kalau yang tadi Down syndrome kan AVSD. And then Turner is coarctation of aorta. Kalau kita dari Marfan, dia elongation of aorta. Yang Noonan nih dia PS ya. So you baca autosome dominan dia. And then another one Dijos syndrome usually TOF ya. Okay, so I think that's all ya. Any more questions? Comment? Alright, so let's close with tasbih kifaran nasr. Alright, thank you all. Assalamualaikum.
Thank you, Doctor.
Thank you, Doctor.
Thank you, Doctor.